All right, hi you guys, I'm back. We're going to finish up chapter 8 here. So I ended at about this spot, I believe, and I'm just going to keep going. Okay, so we talked about delta G, Gibbs free energy, and how biological reactions can happen in the cell, because obviously we need these things to happen. And the two key questions were, can a reaction occur spontaneously? And we answered that question by talking about delta G, or Gibbs free energy. So if delta G was a negative number, that's an exergonic reaction and it will happen spontaneously. And remember, if it was a positive delta G, then it would not happen spontaneously and it would require an input of energy. Okay, but just because, let's just go back for a second and say you've got a negative delta G, you've decided that your reaction can happen spontaneously. The next question would be, is it going to? Will it? Will it occur spontaneously just because it theoretically can? Okay, so that's the big question, okay? The big deal here is that it has to have enough activation energy, okay? So you may remember this from chemistry. I think we talked about it in chemistry. Activation energy is the energy that's required to initiate a reaction. So for example, I was going to burn this piece of paper here in my little bedroom, but I thought I better not. So if you have a piece of paper, it's possible to burn it. I mean, it's flammable. Okay, that doesn't mean that it's going to spontaneously burst into flames. You got to light it on fire. Okay, so that would be the activation energy that's required. So if I had a pencil that I could write on my computer, I would make my graph, which you've probably all seen and which is shown on the next slide. Okay, a reaction's got to have enough energy to get going. That's called activation energy. All right, so here's two types of chemical reactions, energetics. In the first one, we're starting out with some amount of energy right here. This is the energy of the reactants. Let me back up. The y-axis is the energy line, energy level, and the x is the direction of the reaction. So in this case, we're starting out with this amount of energy of the reactants, and it's got to get up to here to have enough energy to then be able to convert to products. So the top of this hill is called the transition state, and this amount of energy is called the activation energy, from the amount you start with to the amount that's required in order to move forward. In this one, we're starting out with this amount of energy, and again, we've got to get over this hump, this energy barrier, in order to have the reaction then proceed to product. Okay, but these are different, right? In this case, on the left, the energy of the reactants is lower than the energy of the products. And in the one on the right, the energy of the reactants is higher than the energy of the products. So then the question is, which reaction is exothermic and which is endothermic? Okay, so exothermic means it releases energy, or it loses energy, or the products have less energy than the reactants. So this is going to be the exothermic one because there's less energy of the products. I should say the products have less energy than the reactants. And so we're talking about delta H now. Okay, so if you did final minus initial, you would get a negative number. So a negative delta H is an exothermic reaction and heat energy is released. This one, the products have more energy than the reactants. So the reaction gained energy. Energy went into the reaction over the course of it, and that is called endothermic. Heat energy went in. So if you subtracted the products minus reactants, you would get a positive delta H, okay? But whether your reaction is endothermic or exothermic, there's some activation energy that has to be overcome to get the reaction to go, okay? And that's just what I said. This one's endothermic, this one's this one's exothermic, and this one's endothermic. Okay, so delta G will tell us if it can happen, and whether you have activation energy or not tells us if it will happen, if it's going to happen. Okay, so all living things need these chemical reactions to happen. Okay, otherwise we would all die. So, how is this going on? How can we get a reaction to occur if the delta G is positive? Okay, we just said that a positive delta G is non-spontaneous and it requires an input of energy. Okay, well, this is called energy coupling. Okay, and this is a huge, huge concept. Okay, so what our bodies do, what cells do, what living systems do is they couple an exergonic reaction to an endergonic reaction to drive it. So they use the energy of an exergonic reaction to make a non-spontaneous reaction go. All right, so that is a very, very big idea. Okay, and let's go talk about ATP for just a second, and then we'll give you an example of a coupled system. This is ATP, this is our energy molecule, adenosine triphosphate. So it's got a nucleotide here of adenine. This is one of those nitrogenous bases, and it's got a sugar. In this case, it's ribose. So this is a monosaccharide, and it's got 3 phosphate groups here. Okay, so this is the structure of ATP. And it is used to drive, I should say the breakdown of ATP is exergonic, and the energy that comes out of that drives endergonic reactions. So for example, here's ATP. It gets broken down, so this is hydrolysis. Here's water. Okay, so it gets broken down. One of these phosphates comes off, leaving ADP. So this is adenosine diphosphate. So there's 2 phosphates. And when that happens, 7.3 kilocalories per mole of energy is released. So when this first phosphate group is broken off, 7.3 kilocalories per mole is released, which can then be used to drive an endergonic reaction. So that's shown again over here. So here's ATP. It's getting converted to ADP plus inorganic phosphate, and another product is energy. So this is our fuel for endergonic reaction. So the breakdown of ATP drives many, many, many endergonic reactions. So it's coupled. So this breakdown reaction will get coupled to reactions that need energy. Okay, so here's an example. Okay, the conversion of glutamic acid to glutamine. So glutamine is just an amino acid. No big deal. This is just an example. And it starts out as glutamic acid, reacts with ammonia, and becomes glutamine. Okay, and that has got a positive delta G of 3.4 kilocalories per mole. So this is an endergonic reaction. It's not going to happen all by itself. Okay, so then if we couple it with the hydrolysis of ATP, that's shown here. So this is, I think, 2 steps, glutamic acid plus ATP. A phosphate from ATP gets transferred to glutamic acid. That's an intermediate. Okay, and then the ammonia comes in and makes the final product of glutamine. And we've also got ADP plus an inorganic phosphate. So this 2-step reaction is the coupling of this reaction with the ATP breaking down to ADP plus inorganic phosphate. Here it is overall down here. So this one requires 3.4 kilocalories per mole. The breakdown of ATP releases 7.3 kilocalories per mole. So when these 2 reactions are coupled, we still end up with a net gain of 3.4 kilocalories per mole, because all of the energy that's released from the ATP breakdown isn't even used by this reaction. So there's some left over. Okay, so this breakdown of ATP is used to drive that endergonic reaction. So that's what we mean by coupling. Okay, and here's another little analogy of ATP and its function. This is a $20 bill. And, you know, it's a nice chunk of change. It's a very useful quantity of money. It's not going to buy you everything. It's not going to buy you a car. You know, it's not going to pay for college, but, you know, it'll get you a cup of coffee and maybe a sandwich. So it's kind of a useful amount of money. It's not huge. So that's kind of like ATP, right? It'll get you something. It'll get something done for you, but it won't achieve everything. Okay, I suppose if you have enough ATP or enough $20 bills, you know, you can do a lot. All right, so let's talk about regenerating ATP, because if we're breaking down our ATP, we need to get it back somehow. Okay, we need to remake ATP. So here on this side, let's look on this side over here. Okay, here's ATP. getting broken down to ADP plus inorganic phosphate. And like we just said, that releases energy, 7.3 kilocalories per mole, and that is used to fuel endergonic processes. But somehow we have to get that remade. So we have to get ADP plus inorganic phosphate to rejoin up, rebond, and reform ATP. So this needs energy from somewhere else. Okay, the big question is, where is that energy coming from? Okay, and the answer is in chapter 9 and chapter 10. Fundamentally, we eat food, okay, and the breakdown of our food supplies energy, okay, and we can regenerate the ATP that's needed for a lot of these biological reactions, okay? Okay, so let me just say this one more time. The energy that's released by the catabolic reactions in the cell, so that's all these reactions of cellular respiration, is the energy that's used to regenerate ATP. So this is actually a really fun unit. You know what? We're going to learn a lot in this unit. Okay, it's pretty great. All right. Moving on. Okay, that is the end of that, and I'm just going to keep on going. Okay. How do we get a reaction to occur if the activation energy is too high? Okay, we got to have enzymes. Okay, I'm sort of assuming you've heard of enzymes, but what they are is they're almost always proteins, and their job is to catalyze reactions and lower the activation energy so that you can make a reaction happen. Okay, so enzymes, you know, changed the order of how reactions happen in order to make them go. Okay, so they're super, super important catalysts. And they're, like I said, we're just going to assume they're always proteins. They're not always proteins, but most of the time they are. This is an analogy of a stick. And again, I don't have my document camera hooked up, but if I had a stick in my hand, an enzyme called stick ACE could break it. So I forgot to tell you that most enzyme names end with ASE. So stick ACE is the enzyme that would break my stick. Okay, my substrate is the thing that the stick, I'm sorry, you know what, I'm gonna get a stick, just a minute. Okay, well, here's the analogy. I ran out to the yard to get a stick. I don't know if it's gonna be really worth it or all that exciting, but let's just do it anyway. Okay, so for enzymes, the substrate is the molecule or the compound that the enzyme is acting on. Okay, so in this case, here's my stick. My substrate is my stick. And then I'm going to call my enzyme stickase. Like I said, most enzyme names end with ASE. Okay, and here's sort of a graph of the energetics. So if I didn't have my enzyme, you know, I would need this amount of energy. This is my activation energy right here. That amount of energy. Okay, so basically, you know, that is a huge barrier of energy, and the stick would never break, you know, without some energy. So if I have my enzyme, it lowers the activation energy. That's the job of an enzyme. And now I only need this much energy. Okay, so this is the activation energy with my enzyme, in this case, stickase. Okay, so I'll just show you how exciting this chemical reaction is. Okay, here's my stick. This is my substrate. I'm going to put my enzyme into it. I'm going to bend this stick. Okay, and when it's almost broken, we're going to call that the transition state. So up here on the top of this energy barrier is called the transition state. And that's where the bonds are distorted or they're just about to break. Usually I think about that transition state as the place where the reactant bonds are all like partially broken and the product bonds are all sort of partially made. So in our stickase example, I got my stick, I'm applying my enzyme, which is me bending it, and I'm going to break it. So now I have my two products. My reactant was my big stick together, and now I have my two products. Okay, so that is the stickase metaphor. And now I can give that broken stick to my good old dog, George. Okay, so back to the slides. Okay, so anyway, that's the basic idea. So for getting reactions to occur, enzymes, as I said, are almost all the time proteins. Okay, so this slide is just a quick review of protein structure. We have the primary structure is the order of the amino acids. The secondary structure is the hydrogen bonding between the between the carbonyl, the C double bond O's, and the amines. We can get these beta pleated sheets. Sometimes you get an alpha helix. This is another kind of secondary structure. Okay, and then we can get this tertiary structure, which is the interactions between the R groups, which you may recall can be ionic, disulfide bonds, nonpolar interactions, or I think I said ionic. I probably forgot one, but that's okay. And then quaternary. Okay, so anyway, enzymes are proteins, and they're going to have your normal protein structure folded up, tertiary structure. Okay, and when they fold, they vary. If they have a job to do, they're going to have an active site. Okay, so inside here, this is obviously a very generic schematic of an enzyme. But the active site of an enzyme is the place where it binds to the substrate. Okay, so the enzyme is going to be able to recognize and bind whatever substrate it's going to act on. Okay, and so in the case of my stickase, it's going to be complementary to the transition state of my stick. So when I had my stick almost bent, okay, the active site of the enzyme, is right at the point of where we're in the transition state. Okay, and there's a different, active site for every substrate. Lots of enzymes are very specific to one chemical reaction. Not all. You know what? There's classes that are. Okay, and then this is what they do. This is kind of what I drew on my paper. If I didn't have an enzyme, we have a much higher barrier, energy barrier, a much bigger activation energy from the starting amount of energy of the reactants to the top of this hill. The job of an enzyme is to lower that. Okay, so with an enzyme, that activation energy is now much lower. We only need this much energy. Okay, they both end up in the same place. The products still have the same amount of energy whether the reaction happened with an enzyme or without an enzyme. Okay, so the final amount of energy doesn't change, just the activation energy. Okay, and you can see that here's the activation energy with the enzyme. Here it is without the enzyme. So this is much bigger. Okay. All right, and then here's a picture of sort of a schematic of a chemical reaction with an enzyme. So here's the enzyme. Okay, here's the substrate. This red piece and this green piece are the substrates. So those are the reactants. Okay, they're binding in this active site of this enzyme. The enzyme then changes the structure of those substrates and changes them into the products, which are in the active site, and then they get released. Okay, so we started out with substrates, we ended up with products, and look what happens here. We get the enzyme back just the same way that it was. Okay, so enzymes themselves end up unchanged. They don't get used up, and their shape is the change. Over the course of this reaction, you can see that this enzyme is undergoing like a shape change. Its conformational change is happening, and that's all part of the process of making these reactants turn into those products. Okay, but enzymes themselves don't get used up. They're regenerated and used over and over and over. Okay, and they come together from random collisions. The enzyme and the substrate have to bump into each other, and they have to hit in the correct orientation in order to make this reaction happen. And that just happens randomly. Okay, there's some things you can do to make it happen more often. Okay, the enzyme changes shape slightly. That's what I'm talking about. So here it's sort of a smooth inverted C, and here it's all lumpy. So it changed shape. That is called induced fit. When the substrate bound into that active site, it induced the enzyme to change shape. Okay, and then I just said this too. So enzymes are catalysts, and they are not used up. They get used over and over and over. A few more things about what affects enzymes. Okay, temperature and pH are super important to enzyme function. Enzymes generally have an optimal temperature and an optimal pH at which they work the best. Okay, so why are these important to enzyme function? Okay, if you have temperature that's too high, you guys know what's going to happen. Okay, your protein will get denatured. Okay, it'll lose its shape. And if your enzyme loses its shape, it's going to lose its active site and it isn't going to work anymore. Okay, and if the pH is wrong, pH is a measure of hydrogen ion concentration, and if you got the wrong amount of hydrogen ion in there, you know, those R groups that maybe need to have a certain charge won't have it and they won't be able to do their jobs. Okay, so temperature and pH are very important for denaturing your enzyme. You know, I should say having the incorrect temperature and the incorrect pH can easily lead to denaturation of an enzyme, which is the loss of its shape. Okay, here's a picture of a nice, happy, folded, properly folded protein. It gets denatured, and you can see that the primary structure is still there. Those amino acids are still hooked together, but it's lost its shape. Okay, awesome. All right, temperature. If it's too hot, yeah, that's when the thing can get denatured. Okay, temperatures that are too cold don't generally, I wouldn't say they denature enzymes, but enzymes don't work if it's too cold because they aren't moving enough. This is the old kinetic molecular theory that we all know and love. You know, we need some heat energy to make things move. So if it's too cold, they're not moving enough, they're not going to collide often enough, they're not going to collide with enough energy, and they're not going to be able to make that reaction happen. All right, and pH is what I just was talking about, how you change the proton concentration, and you're going to shift the balance between those ions, okay, and disrupt the bonds holding the protein together. All right, another important thing is called a cofactor, okay? Cofactors and coenzymes are things that help enzymes work, okay? Cofactors are inorganic, coenzymes are organic, okay? So these are just some examples down here. Here's a substrate. I should back up. Here's the enzyme. Here's a cofactor, so a helper. Here is the substrate coming in to bind, and the reaction happens. And we're going to talk more about these as time goes on, okay? Some examples are iron. Iron would be a cofactor because it's not organic. Remember, organic means it's got carbons in it, and this is just iron. So our blood cells, our hemoglobin, have a place for iron, which really helps it do its thing. Zinc is another one, and then a coenzyme. NAD plus is from niacin. That's definitely got some carbons in there, so that's organic, and those can really help chemical reactions too. All right, inhibitors can decrease enzyme activity. Competitive inhibitors will get in the way of the substrate binding to the active site. So if the substrate is supposed to bind to the active site, but something else is already bound there, you know, the substrate can't get in. Okay, this would be an inhibitor. So a competitive inhibitor is going to compete with the substrate for the active site. All right, a non-competitive inhibitor is going to bind someplace else on the enzyme and somehow change the shape of this enzyme so that the substrate can no longer bind. Okay, so it's going to mess up enzyme function, but not by directly binding the active site. Okay, so competitive inhibitor binds the active site, blocks the substrate. Non-competitive inhibitor binds away from the active site and changes the shape of the enzyme so the substrate cannot bind. All right. Allosteric regulation occurs when protein activity is altered by binding of a molecule to what's called an allosteric site. All right. So an allosteric site is someplace away from the active site. So here's our enzyme. Here's an active site. And in this case, we're going to talk about something that inhibits enzyme function. This inhibitor will bind away from the active site. It's called an allosteric site. Changes the shape of the enzyme, and the substrate can no longer fit, okay? An allosteric activator will bind to the substrate away from the active site. At the moment, the active site can't bind the substrate, but the activator will bind and then change the shape of the active site, so now the substrate can bind. Okay, so that's another way that enzyme function is regulated. Okay, and so it can either inhibit it or activate it. And then feedback inhibition happens when an end product of a biochemical pathway shuts off the process by binding to the allosteric site of an earlier enzyme. Okay, so for example, here's an enzyme, okay, threonine deaminase. Here its active site, and here's its substrate. So the initial substrate here is threonine binding to its active site, goes through this pathway, get all these intermediates that are each catalyzed by a different enzyme, and you get this end product. So these are amino acids, isoleucine. Okay, then isoleucine goes back and binds to the allosteric site on the original substrate. Okay, and now the active site can no longer bind to threonine, and the pathway is switched off. Okay, so here the active site has that shape and it's available for threonine. But this end product of this pathway is isoleucine, and it changes the shape of that active site. So now threonine can't bind there anymore, and the pathway stops. Okay, that is called feedback. All right, and then here's an analogy. You got a whole bunch of stuff plugged in. Okay, that allosteric site is currently occupied by this non-competitive inhibitor, and the substrate can't get in there. All right. Let me see here. I think that I'm going to stop, and we will do these application slides. I think we'll do those in class. All right, awesome.